the University of Alabama, and I'm going to talk a little bit about my dissertation work. So today specifically about what the adaptive value of males are. I think that's a common question, especially in sex evolution, but specifically in a vertebrate that can self-fertilize. So the fish eye study is pretty cool in multiple ways. I'm more concerned with its variation across its distribution. So the mangrove rivulus is found in mangrove forests across Florida, mainland, the Keys, the Caribbean, and a little bit of Central America. It's one of the only two vertebrates that can self-fertilize. It also can change sex from a self-fertilizing hermaphrodite to a male. And what you see here, in this table is what I'm interested in. Across the distribution, there is a lot of variation in the proportion of males, and then it corresponds with observed heterozygosity. So that's our measure of how much outcrossing is happening between the hermaphrodites and males. There isn't any evidence that the hermaphrodites outcross with each other. And so my questions revolve around one, why do they outcross at all? And specifically, what factors cause this large variation? So I'm going to refer back to this graph at the end. This is what we measure as far as the sexual plasticity of each lineage. So on the y-axis is the white portion of a bar is the percent hermaphrodite of that lineage that we have in the lab, so under controlled conditions. And the black part of the bar is of that lineage, the proportion that spontaneously turn male. Under controlled conditions, there's no opportunity for outcrossing. They're kept in isolation. I call this the Marmoratus Marriott. It's very comfortable. They get room service 6 days a week. It's pretty nice. So we have populations from the different areas of the distribution, and these bars represent 359 lineages and almost 30,000 individuals. So again, I'm going to come back to this. This is what represents the sexual plasticity of a lineage, and it ends up being kind of important. So my first question is, one, why does outcrossing occur? And then a sub part of that is, why does it vary across populations? So the obvious hypothesis is that outcrossing gives some advantage to the offspring, particularly in stressful situations. So this is an illustration of Marmoratus Marriott. There's one fish per tub. In my first experiment, I used 69 different genotypes from the Belizean Cays, so island populations, and the Florida Keys. And specifically, I chose lineages that had our full range of heterozygous loci. We measured with 32 microsatellites. And so what I was looking for was some evidence of either inbreeding or potential outbreeding depression across 4 different variables. So we raise our fish in 25 parts per thousand salt water. So that was control. We use a low salinity treatment at 10 ppt. 40 ppt was our high salinity treatment. These are well within the salinities that we find the fish in the mangroves. And they are also a tidal brackish fish, so we had a tidal treatment where it was at 25 ppt, and there were 2 tide intervals per day, high and low tide, that lasted approximately 5.5 hours each. So this was how we controlled the tides. This represents one of these cups. They're in common water tanks so that we can make sure across each treatment the salinity, the temperature, and everything was the same for all of the fish. and then holes in the bottom allowed us to decrease and increase the water level in the tidal treatment. And you can't see it here, but we had replicates of the treatments, and also for the salinity differences, there was a step-down transition tank. We took a bunch of data, so what I'm going to talk a little bit is the mass and length we collected every day from the day that they hatched. Every 30 days, sorry, not every day. Every 30 days, we took them out of the cup, massed them, took a picture for length. We also looked for sex change every week during the egg check. So we can identify if they changed sex from hermaphrodite, which is a brownish gray mottled phenotype, to orange, either freckled or just a solid orange skin phenotype when they turn male. So during the egg checks every week, and then during the measuring, we could see that. We checked for eggs, and then we also measured death, because in our lab, death is data. So in this experiment, we had a little over 1,100 fish that hatched. Today's data, come from those fish that made it into treatment at 90 days post-hatch, and then those that finished treatment at 180 days post-hatch. And then at 180 days, we collected the ones that were still alive, dissected them to measure liver, gonads, all that good stuff. So what we found was that survival was very treatment dependent, which isn't a huge surprise. So in these graphs, the treatments are across the x-axis in the consistent order. This time, this is mortality proportion in each treatment. In high salinity treatment, it was really rough on them. About 50% of the fish died. There was a slightly higher mortality in the tide treatment than than the control treatment, but it was about the same as the low salinity. We measured growth, like I said, every 30 days. This graph represents those that finished the treatment. So these fish were the 672 that lived the whole time. And what we saw was a strong treatment effect of the tide. So they were just not able to put on the same amount of mass as the fish in the other treatments that survived. So, the y-axis is specific growth rate. We checked for eggs every week. So this graph represents all of the fish that we checked for the full 180 days. This does include some of the ones that ended up changing sex to male, because we weren't checking for eggs until we knew they were male. What we found was that in the control treatment and the low salinity treatment had higher fecundity than those fish in the high salinity or the tide treatments. And of the fish that laid at least one egg, we counted their eggs. So the mean number of eggs differed significantly in the tide treatment. A lot fewer eggs, and again, these fish are a lot smaller. They also had a different body shape. You could tell that they were struggling energy-wise. There's also a slight difference in the mean number of eggs out of the high salinity treatment, but it wasn't significant, but it trended down. So overall, what we saw was that the high salinity treatment, high mortality. Those that did survive the high salinity treatment were about the same size as the other treatments, or the low salinity and control, slightly less fecundity though. In the tide treatment, we had a lot less growth and a lot less fecundity. So what about the heterozygosity advantage that we were looking for? No. So I have lots of really high p-values if you'd like to see them, but we didn't find evidence of inbreeding or outbreeding depression. So then the next question is, then why would an animal turn male? You're, as a fish, self-fertilizing hermaphrodite, you're giving up reproductive assurance to become male.
Why?
So, the obvious hypothesis is that to overcome this twofold cost of being male, and you could argue it's even more than 2 here, there must be significant advantages. And that is what we found. So we have proportion that survived the full 180 days here, separated by those fish that remained hermaphrodite versus those that changed sex to male, and overwhelmingly the male survived, almost 100% of the time. And even more interestingly is that male survival was not treatment dependent. So here we have percent surviving into treatment. In the fish that remain hermaphrodites, we see that same trend, where in high salinity there's a lot of mortality. But that is not true for those that change sex to male. They just live. So then we decided to see if there was a difference in the sexual plasticity of the lineage and if it had an effect on survival. So again, this is that graph from earlier. These numbers on the x-axis represent that sexual plasticity quotient that we measure from the colony. So each dot is a lineage, and so it would correspond to like that lineage, 5% of them change sex to male. Again, those that change sex to male, doesn't matter what lineage they come from, they just live. However, with the hermaphrodites, we see a correlation with sexual plasticity and survival. And so the point is, those fish that come from a highly sexual plastic lineage but stay hermaphrodite are at a big disadvantage. They die. So overall, we do have treatment effects. We did not find any heterozygous effects, either inbreeding or outbreeding depression. We did find out that if you change sex to male, you just don't die, which is interesting because a lot of the sex allocation literature is about how to better be reproductive. And you could argue that not dying makes you reproductive. So I would like to thank my funders. I took a ridiculous amount of data for about 1.5 years, and 9 hours a day was what we spent in the fishery. So I had a great group of undergrads, and I'll take questions. Yes?
I'm curious, do you have any idea of how reproductively viable the males are after, say, like the time treatment or anything like that?
After they change sex to male? Okay, so the question is, how reproductively viable are the fish after they change sex to male? I don't know. In this study, they were held in isolation regardless. That is what my next ongoing experiments are about, so come back next year and I'll have that for you. Yes?
I'm really curious about your thoughts on disentangling cause and effect. So is it that this is maleness is condition dependent and high condition hermaphrodites opt for being male, or is it that if you're stressed you can switch to be male to survive better as male?
Right, so her question is about cause and effect. Is the maleness an effect of treatment or vice versa? And we just don't know yet. I mean, that's going to take some distangling with, I guess, different designs, since the experiment was designed to answer something else.
Yeah, super cool.
Yeah.